Neural communication, like muscle excitation, is based on electrophysiology, the cellular mechanism for producing electrical potential and currents. An electrical potential is the difference in the concentration of charged particles between one point and another. An electrical current is a flow of charged particles from one point to another, generally in response to an electrical potential. In the body, currents are movements of ions such as sodium and potassium through gated channels in the plasma membrane. These gated channels are opened or closed by various stimuli. This enables the cell to turn electrical currents on and off. All living cells are polarized and have a resting membrane potential, that is, a charge difference across the membrane. Neurons and muscle cells have become specialized to take advantage of this electrical potential in signaling each other. You've already had a basic introduction to how the synapse works at the neuromuscular junction. In this chapter, we'll go into more detail of how the action potential is propagated along the neuron before the neuron synapses at the muscle junction. A resting membrane potential is usually around negative 70 millivolts in the unstimulated neuron. The negative value means there are more negatively charged particles on the inside of the membrane than there are on the outside. Resting membrane potential exists because of unequal electrolyte distribution between the extracellular fluid outside the cell and the intracellular fluid inside the cell, and it results from a combined effect of three different factors. First, ions diffuse down their concentration gradient through the membrane, but the plasma membrane is only selectively permeable and allows some ions to pass more easily than others. The third thing that influences the resting membrane potential is the electrical attraction of cations and anions to each other. Let's look in more detail. Potassium ions have the biggest influence on the resting membrane potential because the plasma membrane is more permeable to potassium than any other ion. Thus, it leaks out down its concentration gradient until the electrical charge in the cytoplasmic anions attracts it back, and equilibrium is reached. So here we can see that there's a potassium channel. There's many more potassium channels in the membrane. The blue dots are potassium. There's a higher concentration of potassium inside the cell, so it wants to move down its gradient through these potassium channels. Now, potassium has a positive charge, so the balance is reached when potassium diffuses out to a point where not only are we dealing with the chemical concentration coming to equilibrium, but we also have an electrical concentration to figure out. As we lose all kinds of potassium ions, we're losing positive charges out of the cell, and it's becoming more negative. So there'll be an electrical draw for the potassium to move back into the cell. Potassium is about 40 times more concentrated in the cell than out of the cell, so naturally it wants to diffuse out of the cell. However, only so much can go out because of this electrical draw of the anions, the negative charge inside the cell, pulling the potassium ions back in. The cytoplasmic anions cannot escape because they have a very large size or charge. Phosphates, sulfates, organic acids, proteins, ATP, and RNA are examples of negatively charged cytoplasmic anions. They're represented by these green structures here. The membrane is much less permeable to sodium, and sodium is in higher concentration outside than it is inside, so it cannot diffuse down its gradient at such a high rate as potassium can. Some leaks and diffuses into the cell down its gradient. So the resting membrane potential is much less influenced by, in addition to the varying permeability of potassium and sodium channels in the cell membrane, there are also sodium-potassium pumps. You'll probably recall from general biology how they work. 3 sodium move out for every 2 potassium that are brought in. That's more positive charges going out than there are coming in. So we have a net of -1 difference for each cycle of each sodium-potassium pump. Because the sodium-potassium pumps work continuously, they compensate for the sodium and potassium leakage, and it requires a great deal of ATP. 70% of the energy requirement of the nervous system is about fueling the sodium-potassium pumps to maintain this potential difference across the cell membrane. Thus, it necessitates glucose and oxygen to be supplied to the nervous tissue. Although the neuron is said to be resting, it's really not, because when it's not producing signals, it's highly active in maintaining the resting membrane potential and waiting for something to happen. So, here is the first part of our diagramming for this week. Diagram resting membrane potential. I swear it's the easiest way to get an understanding of how it's maintained. First of all, there are anions that are stuck inside the cell, highly negatively charged stuff. Second of all, you've got a variability in the flow of potassium and sodium out of and into the cell respectively because of the difference in the number of potassium channels and sodium channels. There are far more potassium channels, and thus potassium is actually leaking out of the cell constantly. We're losing positive charge as well as having a lot of negative charge on the inside. Now, if it could, sodium would diffuse down its gradient at an equal rate to keep that all in balance. However, there are not as many sodium channels in the membranes, and so sodium can only diffuse in very slowly. As potassium moves out down its concentration gradient, it's going to be changing the electrical potential across the membrane because, after all, it is positively charged. If you have positive charges moving out, then it's making it more negative on the inside. However, at a certain point, that negative charge on the inside is going to act to draw the positive charges back into the cell. So the inflow and outflow of potassium through the channel based on this electrical and chemical gradient will become equal, and thus an equilibrium is reached. This is about -70 millivolts. In addition to these channel differences, as well as the high concentration of large anions, there are sodium-potassium pumps. They account for about -3 millivolts of the cell's resting membrane potential, as they pump out 3 sodiums or 3 positive charges and only let 2 in for each cycle. Thus, we're drawing more negativity into the cell. Spend a moment, put these aspects together in a diagram so that you can gather a good understanding of resting membrane potential before we move on. Now we'll consider some of the disturbances in resting membrane potential that occur when the neuron is stimulated. Typically, the response of a neuron begins at the dendrites and spreads through the soma and then travels down the axon and ends at the synaptic knob. We'll consider this process in order, beginning with looking at local potentials. When a neuron is stimulated by either chemicals, light, heat, or mechanical disturbance, it causes sodium channels to open. This allows sodium to flow in. Remember, there were more potassium leak channels than there were sodium leak channels. Now sodium can rush into the cell. This sodium inflow will neutralize some of the internal negative charge, and the voltage across the membrane will drift upwards towards 0. This is called depolarization. Sodium diffuses for short distances on the inside of the plasma membrane, producing a current that travels towards the cell's trigger zone. This is a short-range change in voltage called a local membrane potential, because it's only changing locally. If there's enough change in local potential, we may be able to elicit an action potential to fire from the axon hillock. But if not, it just remains as a local potential. What's the difference between local potentials and action potentials? Local potentials are graded, thus they can vary in magnitude with the stimulus strength. A stronger stimuli will open more sodium gates. They are decremental, that means they get weaker the further they spread from the point of stimulation. The voltage shift by the sodium inflow diminishes rapidly with the distance it spreads across the cell membrane, much like diffusion of any molecule. Local potentials are reversible. When stimulation ceases, potassium diffusion out of the cell through the leak channels will return the cell membrane to its normal resting potential. They can also be excitatory or inhibitory. Some neurotransmitters, like glycine, will make the membrane potential more negative, that is, hyperpolarize it. It becomes less sensitive and thus less likely produce an action potential. For example, it might open chloride channels, and chloride would diffuse into the cell, making the cell more negative, which would make it harder to reach the level needed to fire an action potential. So graded potentials could lead to an action potential. An action potential is a much more dramatic change that's produced by voltage-regulated ion gates in the plasma membrane. It can only occur where there's a high enough density of voltage-regulated gates. Around the soma, there are only 50 to 75 gates per micrometer. However, in the trigger zone, there are 350 to 500 gates per micrometer. And the action potentials can be generated there because a local potential could be large enough to open these gates and cause a huge influx of sodium. If an excitatory local potential spreads all the way to the trigger zone and it's strong enough when it arrives, it can open all these gates and cause the action potential to be fired and travel along the axon of the neuron. This graphical representation to the right shows us how an action potential might work. It's a rapid up and down shift in the membrane voltage. Here's our negative 70 of resting membrane potential. We're going to see a big voltage shift up towards positive numbers, and then it'll return even further below resting membrane, and finally come back up to resting membrane. The sodium ions that have entered through the channels after stimulation of the soma could flow in current towards the axon hillock, and they can depolarize the membrane at that point. However, you need to have enough stimulus from these sodium ions to reach a critical threshold value, typically about negative 55 millivolts. Once the cell reaches this threshold potential, these voltage-gated sodium channels will be flung open and sodium can rush into the cell. Now, sodium is positive, and so as sodium rushes into the cell, it drives the membrane potential to less negative. The more depolarized the cell becomes, the more sodium channels open. This is a positive feedback cycle that causes a rapid rise in the membrane voltage. As more sodium gates open, more potential change, more gates open, all the way till we get to about positive 35 millivolts. At this point, the sodium gates begin to close, and now we can start returning back towards the resting membrane potential in repolarization. So by the time the voltage peaks, the potassium gates, electrically gated potassium channels. Now these are different than the leak channels that were about resting membrane potential. These are only opening because we reached threshold. However, they're slow. Potassium gates are very slow to open. This gives sodium the opportunity to depolarize the cell. Then the slow potassium gates open while the sodium gates are closing. This allows potassium to rush into the cell. As potassium rushes into the cell begins to become repolarized, or the potential difference across the membrane becomes more negative. Now the potassium gates stay open much longer than the sodium gates. So slightly more potassium leaves the cell than enters the cell. And so we see a hyperpolarization or after potential. Now, in order to return that hyperpolarization back to regular polarization, the sodium-potassium pumps come into play. So during the phases of an action potential that you saw in the last couple of slides, we'll see that sodium and potassium literally exchange places across the membrane. You might ask then, if this is going on constantly, how do we ever get back to normal? Well, remember, there are all these different leak channels that are allowing potassium to leak out of the cell. And we'll also see the action of the sodium-potassium pump constantly reversing this. So it's important to remember that it's only a thin layer of cytoplasm right next to the cell membrane that's being affected. So even though these figures might lead you to believe the whole cell on the inside is seeing this exchange, it is only a thin layer right next to the cell membrane. So really, very few ions are actually involved. Because of this threshold phenomenon, the action potential is an all or none event. If threshold is reached, then all the action potential gates are flung open. Sodium rushes in, potassium rushes out, and we'll see firing at a maximal voltage. If we don't reach threshold, then there will be no action potential. Action potentials are non-decremental. That means they do not get weaker with distance like local potentials did. And they're irreversible. Once we reach threshold, it's all go. So do note the differences between an action potential and a local potential. So now, it really is diagramming time. This will be the basis of your next diagram. What happens in each phase? Let's look at an animation to recap and help us understand how an action potential works. After all, picture is worth 1,000 words. A moving one? Whoa, way more. So in this animation, there's a little bit more detail than we've covered in our text. It involves the sodium activation and inactivation gates. I think that part's kind of neat, so you can add it in there when you do your diagram. But here goes.
When the cell membrane is at its resting membrane potential, the activation gates of the voltage-gated sodium ion channels are closed and the inactivation gates are open. Voltage-gated potassium ion channels are closed. Depolarization is initiated by a stimulus which makes the membrane potential more positive, causing the voltage-gated sodium ion channels to start to open. As threshold is reached, many sodium channels open. Sodium ions diffuse across the membrane, causing depolarization. Voltage-gated potassium ion channels also begin to open, but more slowly. Therefore, depolarization occurs because more sodium ions diffuse into the cell than potassium ions diffuse out of it. As the membrane potential approaches maximum depolarization, the inactivation gates of the voltage-gated sodium ion channels begin to close, and the diffusion of sodium ions decreases. The potassium ion channels remain open, and potassium ions continue to diffuse out of the cell. The increased potassium ion permeability lasts slightly longer than the time required to bring the membrane potential back to its resting level. The extra efflux of potassium ions causes the membrane potential to become slightly more negative than the resting value. After the voltage-gated potassium ion channels close, the active transport of sodium and potassium ions reestablishes the resting membrane potential.
That shows us what's going on with these gates. You want to put this information along with the graph of an action potential and show what's going on during an action potential, what's going on in the depolarization phase, what's going on during repolarization. How do we actually reach threshold? It may take you some effort to put this together. By all means, have some discussion on the discussion board about these concepts. It's tough stuff, but once you get it, it's really cool.